Okay, we're going to continue, pick up where we left off yesterday. I think this was the slide that we had ended on. Discussing the different classes of macromolecules that we're going to get into for the rest of this chapter. The two we're going to cover today are carbohydrates and proteins. They seem to be like they might be the two most lengthy ones that we'll get into, so we'll get the long lecture out of the way first, and then lipids and nucleic acids will be a little bit shorter on the back end. Okay, so we will go through carbohydrates first. The first slide that we're going to see here just describes some of the basic stuff about carbohydrates, some of the stuff you might already know. The fact that it is called a sugar talks about its namesake, the terms carbo and hydrate, meaning carbon and water, basically give you a really good understanding of its generic molecular formula. Okay, anytime we see any type of molecule that has C, H, and O in this ratio like we see here, we can think of it as being a carbohydrate. So we're going to see quite a few examples of that. If you really wanted to, you could go break down what their structural formulas are into their molecular chemical formulas, and it would yield some kind of ratio like you see here, that there's twice as many hydrogen for either C, or carbon, or oxygen. Okay, and if you look even closer at this, and I had to look at this a couple times before I was able to even recognize it, that term carbo and hydrates, carbon and water together, is just that, is a carbon together with a water. Cool, huh? Okay, so we see the term carbohydrate has a little bit more meaning. And then we get that ratio of hydrogens to 1C and 1O. Now, it does, we are going to cover the functions of these macromolecules, these larger macromolecules, as we work our way through them. Some of the macromolecules have more functions than others, and some of them I'll spend more time on than others. Others have a few less, but I will mention some of the functions kind of intermingled as we go throughout all of the material on each one. So some of the general functions of carbohydrates is that they help to store energy in chemical bonds. You know, when you think about sugar, oftentimes we think of sugar as energy stored in chemical bonds, right? It's the sun's energy stored in chemical bonds. There's more to that story, it turns out, but that's for a biochemistry class, which we're just not, this is a biochemistry chapter, not a biochemistry class. So we'll kind of leave it at that to let you know that there is more to this idea of energy being stored in bonds or in molecules. Okay? Glucose tends to be the most common monosaccharide. Okay, that's because plants make it, and plants are one of the most numerous things on the face of the planet. And one of the most common basic sugars or carbohydrates is glucose, and it is produced by photosynthetic autotrophs known as plants. So the way that we're going to address each one of these, or I should say most of these macromolecules, these large macromolecules like carbohydrates, is we're going to start with the monomer structure, the singular subunit. And then we're going to talk about those the properties of those constituent parts. We're going to talk about what happens when we put those parts together into larger polymers, and we'll start to talk about some of the functions and the properties of those larger molecules as a whole. Okay, so the structure of monosaccharides: it has an OH group that is attached to each carbon except one, which is double bonded to an oxygen. Pardon me, just about drooled everywhere. And that's called a carbonyl group, and we see that carbonyl group up at the top here. It's not necessarily that you need to commit this to memory, but it's characteristic of all monosaccharides. Okay, and then here we see attached to all of the carbons, aside from that one, are OH groups or alcohol groups or hydroxide groups. Okay, that is kind of the generic structure of most or all monosaccharides. Okay, oftentimes we can have different lengths of monosaccharides, which ultimately have different properties based on how many carbons they have. Here we see the very beginning one. We see one called glyceraldehyde. That is a triose. It's a triose sugar. It's got 3 carbons. This is going to become famous for us when we get into chapter 9 and 10. Glyceraldehyde is something that we'll see again in those chapters. Ribose is a pentose sugar, or a sugar with 5 carbons. Ribose is the specific name of this specific one because it has this specific structure. There are other 5-carbon sugars. For instance, fructose is an example of a 5-carbon sugar. It has a different structure than ribose, but it is a 5-carbon sugar. Then we have these things called hexose. Okay, hexose is the name of 6 carbon sugars, and we've got multiple different kinds of 6 carbon sugars as well. We've got glucose and galactose. They look very, very similar, aside from just one change in their structure. Okay, so glucose and galactose you could think of are isomers of one another. Okay, and we talked about those in chapter 4. All right. In aqueous solutions, many of these monosaccharides form rings, and I think that's important to address here, because we hardly ever see monosaccharides represented in this conformation here. And that's because most of the time when we're dealing with monosaccharides like glucose, they're in aqueous solutions. They are found in animals. They are found in plants. They are found mixed in with water. So the form that we saw on monosaccharides in the previous slide, like this linear form, oftentimes we don't see that in nature or when it's under study. Oftentimes it folds up in this aqueous form and creates this ring structure. Okay, now I think that's important to understand here, because what we will ultimately see in the book and some of the diagrams, we will see these ring structures without all the letters and the numbers on them. And you have to understand, to think to yourself that that is a monomer structure of a sugar. Okay, and so that your mind should automatically pop to, when I see these very distinctive rings, I'm going to automatically think it's a sugar molecule. Okay, so just keep that in the back of your head. All right, disaccharides. Disaccharides are double sugars. They are 2 sugars. We don't see a lot of this kind of di structure in some of the other larger macromolecules. Usually it's you've got the monomer and they come together into these big polymers. But we do see this very distinctive di structure with sugars. Okay. One thing I neglected to mention in the previous slide when we talk about monosaccharides is another way of identifying these things by their name, and the fact that the name can be descriptive. And it's also something that we'll see in terms of word root kind of word play that will pop up in other areas of our study. Okay, this term disaccharide. Let's break that down real quick. The term di means 2, and then whenever you hear the term like saccharide, okay, that stands for sugar. So 2 sugars. Disaccharides are 2 sugars put together. Monosaccharide, if you go back to the previous slides, monosaccharide is a single sugar. Okay. So disaccharides are double sugars that consist of 2 monosaccharides joined together by something called a glycosidic linkage. Now, in the previous video, I had mentioned something called a covalent linkage, which is basically just a fancy term for a covalent bond. This here is a fancy term for a sugar bond. Okay, so if I had a writey utensil, a thing that writes here, I would write sugar bond. Okay, it's a bond between sugars. And you'll notice that when we get to some of these other macromolecules, that we will start to see descriptive terms for their bonds as well. So do keep that in mind. They do have descriptive terms for the bonds. This one is called a glycosidic linkage. So what reaction forms this bond between 2 monomer structures? Okay, here's a monomer structure, here's another monomer structure. What forms the bond between those? What reaction forms that? I'll give you a hint. We learned about it in the previous video. It's called a condensation or dehydration synthesis reaction. Okay? And if you could take that very generic diagram that I shared in the PowerPoint's video yesterday or the day before, you will notice that it looks almost just like the diagram that you see in front of you right now. Okay? You've got a monomer structure right here in glucose. You've got another monomer structure right here in glucose. And you want to join them together to make maltose, which is a disaccharide. You've got to perform a condensation reaction. And we see that happening right in between those two. I'm going to put the pointer right there, the red pointer right in front. So we see the OH from the glucose on the left combining with the H from the glucose on the right. They form water. The water falls out of the reaction. Okay, it condenses. It leaves that reaction. It's gone. It goes to do something else in the cell. We don't care what it is right now. And what we see as a result is right here. Okay, we see right there, it's the oxygen right where the pointer's at. We see that oxygen is actually this guy right here that is left over and is then linked to this left side glucose over here. Kind of a neat reaction. But we see that representation of the condensation reaction we talked about in yesterday's video, represented right in front of us in a more specific reaction between two glucose molecules that form a maltose, a disaccharide. Okay? Now there's a couple of other examples of disaccharide. There's one called lactose. You guys probably know where you find lactose. It's in milk. Some people are intolerant of this. And what that means is that you don't have the proper enzymes or maybe enough enzymes that will help break down this lactose, okay, break it in half, okay, so that your body can digest it. And we've got one called sucrose. If you know anything about different types of sugar, this is a very, very common one called table sugar. Your table sugar you put on your to bake with, or you put it on your cereal, put it in your coffee, that is sucrose. Okay, so this is an example of sucrose. You don't have to memorize these. I just like to throw them in there and talk about experiences with each one, but we're going to motor on through. But those are examples of disaccharides, and they are put together, either way you cut it, by condensation reactions. All right. The next one we're going to talk about a little bit more in depth and a little bit more at length is the polysaccharides. Okay, polysaccharides are, let's break this word down too since we're here. Poly, we should know, means many. Saccharide again means sugar. Okay, we can think of many sugar units put together makes a polysaccharide. Okay, so the structure of polysaccharides can be varied. It can change from a few hundred to a few thousand monosaccharides all linked together. They can be in a spiral, kind of almost like a helix form. They can be very structured. They can be branching. So the structures of these polysaccharides can be very, very different depending on the molecule that you happen to be talking about. Okay? Primarily, polysaccharides are used for energy storage or for structural support. Okay? And what we see is we see these polysaccharides represented in both plants and animals, but the structure of each in the different organisms is different. Okay? So, for instance, let's talk about glycogen and starch first, because glycogen and starch have analogous functions. They both function for energy storage in their representative organisms. Okay? Glycogen is the energy storage form of sugar for animals. Starch is the energy storage form of sugar for plants. Okay? So they do the same thing for the most part, but they are put together differently. I don't know why. I'm sure there is an answer for it. Off the top of my head, I don't know. I'm sure there's some reason for it. I'm sure it's something I could probably figure out if given enough time. I just don't have enough time right now. Okay? It'd be a good thing to Google if you want. Go ahead and do that. But anyway, we see glycogen is glucose molecules that are put together in this highly branched chain of sugars. Okay? And ultimately what they turn into are these little granules. Okay? And those little granules are actually stored in the liver. In chapter 11, we're going to talk about the release of these sugar molecules under given amounts of stress. Okay, it's a really cool little thing. So we will revisit this, and I will remind you of that when we get to that stuff in chapter 11. But glycogen this is what it looks like. It's a highly branched storage form of sugar for animals, and it's stored in the liver in little granules. Starch is found in the chloroplasts, and it is the stored form of sugar, and we can see here it's almost like a staircase, like a spiral staircase. Okay? They do, for the most part, the exact same type of thing. If the plant needs extra sugar for whatever reason, it'll break the starch down and it will utilize it. If the animal needs sugar for whatever reason, it's run out, so got low blood sugar or whatever, it will access any of the glycogen that's in the liver, it will release that, and it'll get some excess left that's let out for it. Okay? All right, let's talk about the structural forms of these sugar molecules, these polysaccharides. We'll talk first about cellulose, and then I'll transition to the next slide and talk about something called chitin. And so cellulose is a structural molecule that is found in the cell walls of plants. Okay? Plant cell walls are very, very rigid. Okay? And the reason that they're very, very rigid is because of the way that these cellulose fibers are put together. I know this gray thing kind of gets put in the way, but if you look right here, there's a long string of sugar molecules put together in a chain, kind of. They're just a linear chain. And then you've got two more that are right next to it. These molecules are actually probably attached by hydrogen bonds in some way. These long linear string-like molecules are attached. And they're also bundled into something called a microfibril. Okay? And a microfibril is a very rigid structure that is found in the cell wall to actually create that rigidity that you see in there. Okay, it's very, the structure of the cell wall, those fibers are very linear, they're very structured, they're very put together, because that is what we see in terms of the best design for structural supports and things. In fact, these cellulose fibers look a lot like rebar. If you guys know what rebar is, they put rebar in concrete and of buildings. It's like long, thin pieces of metal that are embedded in the concrete, and it provides structure and support. And usually when they put this rebar in, it's either in like a grid pattern like this, or they're laid side by side parallel with one another, okay? And it allows for certain amounts of rigidity, but also some flexibility as well, okay? So strength and flexibility. And we see this with cellulose, okay? Now, cellulose happens to be one of the things that our bodies are not very good at digesting. In fact, we're really, really bad at cellulose. And this is where we would, if we were in class, we would go off on some tangent. We'd stray from our path for a while, and we'd talk about poop. Because I would ask some people in class, hey, how do we know that cellulose is not digestible by humans? And then I'd ask for, examples of what you see in the toilet, because everybody looks at it, right? You all look at your own poo. I do. I'm going to be upfront and honest with you. I look at my own poo. I've seen my kids poop. And there's stuff in there, right? And not all of it gets broken down. You eat a lot of salad one week, your poop has a lot of salad in it. You eat, here's the fun one, corn, right? Everybody knows that corn comes out the other end almost looking the exact same way as when it went in, right? And that's because the corn, the kernels of the corn, the yellow part, is made of cellulose. It's highly cellulose based. And that's the reason it comes out. Like your body can't digest it. And it begs the question, why do we eat corn? Okay? We eat corn because it tastes good. Well, what makes it taste good? I always bring that question up. And the answer to that is it's full of sugar, right? It's sweet corn, right? You eat sweet corn because it's sweet and it tastes good and you like the taste, right? And so what your body does, your teeth can basically break open that yellow kernel casing that is surrounding the starchy material inside of the kernel of corn, and then the enzymes in your mouth and your saliva kind of dissolve it, and then you taste it, and it tastes good, right? But your body spits out the rest of the crap on the other side. Now, one thing to watch out for when you have kids, because it scared the crap out of me, not really pun intended, but scared the crap out of me when I first saw this. My kids, when they were little and still in diapers, loved watermelon, and they still love watermelon, but little kids, when they poop out watermelon, it looks like they've crapped out their insides. It looks like there's bloody intestines in their diaper. And you look up at them while you're changing the diaper, and you're like, you're smiling right now. You're not hurting at all. And it turns out that a lot of the fibers inside of the watermelon kind of keep that red color, and it comes out the other end looking really weird. So, all right, poop talk's done. Let's go on to the next slide, shall we? This next slide kind of gives a general overview of the definitions of these things, what they're used for, what their structure is a little bit like. With regard to chitin, we haven't really talked about that one yet. Chitin is a polysaccharide that's used as a structural material in arthropods' exoskeletons and also in some in fungal cell walls. So the exoskeletons of a lot of bugs are made up of chitin, which is a polysaccharide, a highly organized, very strong polysaccharide. So the chitin that we see here is analogous to the cellulose. Okay, the glycogen is analogous to the starch. Starch and cellulose are plant-based. Glycogen and chitin are animal-based. Okay. All right, let's jump into proteins. We'll go right into it. No screwing around. We're just going to power through, and hopefully this lecture will be done in less than an hour-ish, okay? That being said, proteins are probably the most important macromolecules that we'll touch on, at least today. It's the one we tend to spend most of our time on. It's the one we tend to see repeated over and over again in the material. Proteins are also known as polypeptide chains or amino acid chains or polypeptides. Okay? All of those things, I will use those terms interchangeably, and I want to make sure you understand that so that when I'm just talking, you understand what I'm talking about. Okay? Because sometimes I'll just kind of seamlessly, without really knowing it, call one thing an amino acid chain, and then the same thing I'll call a protein. They're the same thing, okay? So, proteins consist of peptide bonds between 20 possible amino acid monomers. Okay, so the same thing is going to apply here. We're going to talk about the monomers of proteins. We're going to talk about the structure and some of the properties of those monomers, and we're going to talk about how we put the monomers together and how we make bigger structures out of those, and then we'll talk about some of the functions of some of those bigger structures as we work our way towards the end. When we put these amino acid monomers together in a much larger polypeptide strand, ultimately we get a three-dimensional blob or globular shape. Okay, and an example of a three-dimensional globular shape in terms of a protein is shown in that picture there. Okay, these things they look like big old blobs once they're complete. Really, a protein doesn't have much function until you give it that three-dimensional structure. Okay, and that's something that we're going to notice as we work our way through the structures of these proteins, is that not all proteins at all stages of their development or construction are three-dimensional. There are stages at which they're just two-dimensional, okay? Or they kind of lay in a flat string. All right? So the polypeptide is called such because it consists of many peptide bonds, okay? Polypeptides consist of many peptide bonds. So the term peptide bond is the very descriptive way of telling us what kind of bond connects amino acid monomers. Okay? So let's talk about the connection of amino acids first. Okay? The amino acid monomer has a very systematic or a very stock construction. Okay? Amino acid monomers consist of an asymmetric carbon, which is the carbon in the middle. That's the way they describe the carbon in the middle is asymmetric carbon. All amino acid monomers have a hydrogen. They have an amine group or an amino group, whichever you want to call it. And they also have a carboxyl group or a carboxylic acid group. That's the namesake that I talked about in Chapter 4. The namesake of the amino acid comes from the fact that all amino acids, all of these single monomers, have an amine functional group and also a carboxylic acid functional group. Okay? The fourth thing that attaches to the central asymmetric carbon is known as the variable R group. Okay? Each of the 20 specific amino acids has a different variable R group. Okay? It is the one thing that is distinct between all 20 amino acids. I've given you a couple of examples right here. Glycine, if we look at the structure of this amino acid, this is glycine, it has that central carbon, it has the carboxyl group, carboxylic acid group, it has the amine group, it has the hydrogen, but down here is its variable R group. That's the thing that could feasibly change. Okay? So let's compare it to alanine on the right side there. Alanine also has the amine group. It has the central carbon. It has that H on the top. It has over on the right side, it's got the carboxylic acid group. But where it differs is in that CH3 functional group. That's called a methyl group. Okay? So alanine has a methyl group. Glycine is the simplest amino acid that we can get because its variable R group is an H. In alanine, we've taken that H off and we've exchanged it with a CH3 group. Okay? Some of them are very, very simple. Some of those variable R groups are very simple, like the ones you see in front of you. Others are very, very complex and very, very big. Okay? Now, the variable R group is important, so I want to spend a little bit of time talking about the variable R groups. The variable R groups impart properties to the amino acid as a whole. Okay? So if we're thinking about the properties of organic molecules, the R group is responsible for giving a specific amino acid a specific set of superpowers. Okay? I liken these, I don't know, I don't play video games much, but I have a lot of students who play video games, and they talk about Call of Duty and things like that, and there's a new Call of Duty out apparently, which is really fun. But anyway, you get these perks, right, which are kind of like superpowers, or they let you do certain special things, and that's kind of what these variable R groups do. If you have a certain variable R group, that means that the molecule that has that specific R group is going to have a certain set of chemical properties. Okay? So some examples of those are listed below. The amino acid R groups are grouped by polarity. Okay? Some of them are polar, which means they're going to create an amino acid that is water soluble. Some of the variable R groups are nonpolar, which means they're going to create an amino acid that is water insoluble or not soluble in water. Some of the variable R groups are charged, while others are negatively charged, and that has implications on whether or not they can dissolve as well, okay, or what type of things they interact with and how, whether or not they might make hydrogen bonds, for instance, okay. So the examples you see down here, there's a couple of examples you see down here in the polar ones where all of a sudden we notice, oh wow, those variable R groups highlighted in like peach or salmon or pink or whatever you want to call that color, they're starting to get big. It's no longer just like an H or a CH3, it's a CH2SH, or it's a CH2CH, CH3, CH3. Oh my gosh. But the long story, or the short story here is that depending on what the variable R group is, whether or not it's polar, like the one we see in cysteine, it has that CH2SH group, that little pink area, that variable R group, is actually polar, which makes the entire molecule as a whole polar as well. Now if we look at the one next to it, leucine, okay, leucine is nonpolar, and the reason we see that is because it's got a lot of hydrocarbons. Hydrocarbons, if you recall, are nonpolar things. Okay, so that little peach shaded area that we see for leucine is nonpolar because it's a big hydrocarbon, which makes the entire amino acid of leucine nonpolar. Okay, and so we see that same thing here. There's a positive charge over here on lysine, which is my favorite amino acid, and negative charge here on glutamic acid. All right, so that is a discussion of the monomer, okay, and the properties of the amino acid, which is the monomer of a protein. Okay, now we're going to start to talk about how we put those monomers together into a much larger structure, a much larger, more complicated, more functional structure, we should say. Now, this process is a lot more complicated than it is in any of the other three large macromolecules of life. Okay, protein structure is broken up into 4 levels of construction and folding. Okay, the primary structure is nothing more than us connecting amino acids together. I'll give you 3 guesses as to how we would connect 2 amino acid monomers together. I hope you only need one because we've talked about it. It is the condensation reaction, right? We use condensation reactions or dehydration synthesis reactions to form peptide bonds between amino acids. Okay, so if we look at the diagram on the left here. The diagram on the left shows 2 amino acids that we want to try to link together. We want to try to link them together. Amino acid 1 has an O or an OH. Amino acid 2 has some random H's here from the amines. These 2 things are going to combine to form water. The water leaves, right? That's the stereotypic thing that happens in a condensation reaction. And as a result, we see highlighted at the bottom part of that picture in yellow and drawn in red the peptide bond. Okay, the peptide bond is what forms when we have the water drop off of those 2 molecules. We get those 2 amino acids linking together. Okay, so we see the peptide bond over on the right side as well. So the primary structure is nothing more than a bunch of amino acids. Oh yeah, I want to make sure that I link this video because it's great. The primary structure is nothing more than amino acids that are continuously linked together. Now what we see right here is a string of amino acids. Disregard the 2 S's on the side, in the middle I should say. We'll talk about those later. But we see here a string of amino acids linked together. We've got glycine, isoleucine, valine, cysteine, glutamic acid, glen. I don't even know what all of them are. Yeah, I know a lot of them, but you don't have to memorize all of them. You'll naturally, the longer you play around with them, the more you learn them. Anyway, we see all these little singular monomers linked together, and in between here, this would be like a peptide bond, and here would be another peptide bond, and another one. This entire structure is what we classify as the primary structure. It's nothing more than a ribbon or a string of amino acids linked together. Now, the one thing I do want you to pay attention to is the ends of these things. Very similar to what we saw in the very first video. We see that there is an open NH3 or an open NH2, an open amine group here, and an open carboxylic acid group here. And so what that allows us to do is to continually add on at either end, depending on what our DNA sequence is. The DNA sequence is what governs why this looks the way it does. Okay, so based on our DNA sequence, we could continue to add to either side of this amino acid strand based on what we need. Okay? All right. Next we have what's called the secondary structure. The secondary structure is where we start to fold this amino acid chain, this protein. And what we get is we get these repeated foldings of the polypeptide strand that we saw on the previous slide. Okay? And the folds that we get in this particular type of structure, this secondary structure, are stabilized by hydrogen bonds. Okay, we talked about the importance of hydrogen bonds already. I said that they would continue to pop up, especially when we get into large macromolecules, and they do. Here's one example of where we see them. So this secondary structure is stabilized by hydrogen bonds between different amino acids that we see in the chains. Okay, that'll make more sense when we see a different view of what these structures look like. Now, characteristically, the secondary structure is defined by what the folding ends up looking like. Okay, the folding can end up looking like what is known as an alpha helix, and that's what we see over here. This is a 3-dimensional ribbon model of what an alpha helix would look like. Or it can look like what is known as a beta pleated sheet. Okay, and in terms of this computer animation or computer cartoon, it looks like just a flat ribbon. Okay. Now, the picture we'll see next gives you a little bit more indication as to the role that H or hydrogen bonds play, and also where we get this term pleated sheet. Okay. The reason that computers spit out an image that looks like these right here, like you see on the diagrams here, is because it's more pleasing to the eye. Okay, but in actuality, the pleated sheet looks like a pleated sheet. They wouldn't call it that if it didn't, right? The pleated sheet looks like a sheet, like so you take a piece of paper, like when you were a little kid and you make like the fan. Like I'm gonna make a little fan right here, as if this school isn't cold enough right now. My room's like 60 degrees. Okay, so that's a pleated sheet. I just made a little fan, and if I wanted to fan myself, I could do such, right? But it looks the same as what you see on the diagram in front of you. And based on the genetic code of a particular individual or a particular protein, you will get a structure that has some pleats in it, some sheets in it, some ribbon that looks almost like it's been folded. Okay? Or you can get some areas of your polypeptide strand that looks like this helix. It looks like a spiral staircase. Okay? Now let me try to correlate this diagram, which I think is a very important diagram, with one that you've already seen. This ribbon right here, okay, this purple ribbon that you see right here, nice flat ribbon, it's not folded and it's not twisted on itself, is the primary structure of this diagram. Okay? This part right here, if I could draw on it, would have an amino acid right here, and another amino acid linked right here, and another amino acid linked right here, and another one linked right here. Okay? That is the same as what we see in this diagram here. Okay? It's just a different way of looking at it. And so what we see is that this protein, this primary structure, is going to start to fold on itself. Okay? Right now it's still just primary structure. We're going, I need my glasses to zoom in on this. Hold on. We're going around here, okay, we got to this part. Okay, primary structure. All that stuff I just went through is primary structure until we get to this point here. Now all of a sudden it starts folding on itself, and the reason for that is because of the chemical nature or which amino acids happen to be in that particular area at that particular time. Okay, we go here, okay, here's some more primary structure, here's some more primary structure, more primary, more primary, more primary. Oh, here's some more pleated sheets. Okay, eventually we get to the point where we get to this point here. It's an alpha helix. Okay? And so the reason that this alpha helix and these beta pleated sheets are kind of intermingled in this primary structure is because of the association of different amino acids with neighboring amino acids. Okay? This right here is the carboxyl group of this particular amino acid, okay, or pardon me, this particular amino acid, and it has a slight negative charge, and it can make a hydrogen bond with the H from this amino acid down here. Okay, so they're in close enough proximity where you will have the slightly positive hydrogen interacting with the slightly negative oxygen from another adjacent amino acid. Okay, so it causes this structure to form, which is known as the alpha helix, because of these close association where hydrogen bonds can form. And that's why we see over here, that's why we see these folds, these pleats in this ribbon, is because there is some kind of close association between an oxygen with a negative and a hydrogen with a positive that causes some kind of folding. Okay? All right, let's continue here. The next thing we're going to talk about is the tertiary structure. The tertiary structure is where we start to get some functionality between with our proteins. Okay? Tertiary structures have irregular contortions of a protein due to bonding between R groups. Okay? So in the previous one we saw, we get these secondary structures because of hydrogen bonding between adjacent amino acids. We get further folding due to bonding between R groups. Okay, so there is interaction between these variable R groups that I said were important in the monomer structure of the amino acids. Okay? Now these bonds can manifest in a couple of different ways. They can form weak bonds or sometimes strong bonds. Oftentimes it is weak bonds, things like hydrogen bonding. So we can still have hydrogen bonding between R groups. We can have ionic bonding between charged side chains, between charged R groups, because there are those things. We can have hydrophobic and van der Waals interactions between highly nonpolar side chains. And then we can have what are known as disulfide bridges, which form very strong covalent bonds. Okay? So let's look at another diagram here. This is just another way of looking at the same thing. Again, this purple ribbon that we see right here is the primary structure. You could think of little dots all along this thing, and if I had a working pen or something, I could draw these little dots all along here. And these things that shoot off into the middle space here, this right here is a variable R group that would be attached to an amino acid right there where my pointer's at. This big chunky hydrocarbon thing right here, this CH3CH3CH, is the R group of that amino acid right there where my pointer's at. Okay. This CH2S functional group would be attached to that point right there. This big long CH2, 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 NH3 plus would be attached to the amino acid where my pointer is right here. Okay? So what this diagram is meant to show you is that the variable R groups will interact. And when they interact, they create a very distinct three-dimensional shape. This, the diagram that you see in front of you right now, only comes together in this very specific folded conformation because those very specific amino acids have those very specific variable R groups in that very specific space. If you haven't noticed, it's very specific, okay? And what accounts for that specificity is your genetic code. The reason that this polypeptide chain that you see in front of you folds the way it does is because it's coded for in your genetics, which I think is absolutely fascinating, that those little letters that you have in your DNA cause this protein to fold in the most specific of ways, because if it doesn't fold in this way, it doesn't work. Okay? If there is some kind of change in the way it's folded or some kind of mistake in the way it's folded, we're going to talk about some of those mistakes, right? You get a malfunctioning protein. And when that protein malfunctions, you could die. Okay? So, don't want to put it on you that much, but that's kind of the implications of it. Anyway, these R groups have interacted, and they've created some type of bonds. Okay? So here we see the different types of bonds. This R group right here creates an interaction with this R group here that makes a hydrogen bond. This big chunky hydrocarbon side group here interacts with this big chunky hydrocarbon side group here in something known as van der Waals interactions. I'm pretty sure that they talk about those in AP Chem, right? It's almost like they're kind of sticky. They attract each other. Disulfide bridges are the one strong bond that can be formed here. Okay, ionic bonds. You got this kind of weaker interaction here where there's negatively charged oxygen here and a positively charged NH3 here, and they form an ionic bond, okay, which is distinctly different from a hydrogen bond. So, these side chains, these are responsible for the tertiary three-dimensional folded structure. Maybe if somebody really wants to ask me, I don't want to risk like losing the rest of this video going off on a tangent, but if you really want to ask me about hair and biology, I got a great story about Mr. Christofferson's daughter and her mother doing her hair. It's great. So if you want to ask about that, we can talk about it on one of our synchronous sessions. All right, quaternary structure. This is the fourth and final level of protein folding. This is where we take at least two, if not more, tertiary structures, and we smash them together into one giant amalgamation. I liken this to something from my childhood and also an example from yours. If you've ever heard of Voltron, and Voltron was like the greatest cartoon when I was a little kid. It's like all these robot panthers or whatever, and they come together. So singularly, they're panthers that go fight, evil or whatever, and then they can come together into this giant robot called Voltron. That's kind of the way I think of the quaternary structure of protein, of a protein. Analogous example for you guys, in mine too, is the Power Rangers. They kind of have something similar going on. They all have their single robots and they come together into one supreme robot or whatever. But quaternary structure is that. You take these tertiary structures, okay, and you put them together, multiple tertiary structures, put them together, you form the quaternary structure. Oftentimes, this is the completed, 100% done functional form of a protein, okay? So we started with the primary, folded it a little bit. We got some of those alpha helixes. We got some of those beta pleated sheets. We folded it even more with interactions with the R groups, and then we smash all those like three-dimensional structures together into the quaternary. Okay. The example you see here, it's a couple of examples. Collagen takes these polypeptides, these 3 polypeptide tertiary structures, puts them together into this kind of, almost looks like a highly organized structure, and it is. It's a structural protein, so it makes sense that it's highly organized in its structure. The one we see over here is a hemoglobin molecule. It's actually made up of 4 different subunits, and you put them all together, and you get a functional hemoglobin molecule. If one of them is missing, then it doesn't work the way it should. So we see at least 4 different tertiary structures put together here. We see this orange beta chain. We see this kind of purplish alpha chain, these other 2 brown and pink chains over here. You put them together, and then your blood can carry oxygen. Kind of a nice option to have, right, if you want to stay alive. All right, so I really like this diagram as a way to study all of the different ways of protein folding. Okay, it gives you an example of what the primary structure is as a ribbon. Okay, and it even throws in a couple of the abbreviations of the amino acids. Then it shows you the secondary structure, the 2 different interactions there of adjacent amino acids to show the secondary structure. Here it shows all of that together. It shows some secondary structure here in the alpha helix, shows some secondary structure here with the beta pleated sheet, all kind of mashed up into this blob called the tertiary structure. And that blob is held together that way because of the interactions of the variable group R side chains. Okay, and then we take one of these big blobs that we see here, put it together with a bunch of other big blobs, and you get your quaternary structure, which is your fully functional protein. Okay, so where do we go from here? Certain things determine protein conformations, okay, or protein folding. Okay, number one, I guess I probably should change this to put it official. Your DNA factors into how your proteins fold, what the protein is going to look like. This process occurs during protein synthesis within your cell. It depends on the physical conditions of the environment. Okay, the pH can change what your proteins look like. The temperature, the salt, all of these different things can actually change what your proteins look like. pH can affect your hydrogen bonds. Okay, temperature can affect whether or not your bonds are broken. Right, if your bonds are too hot, they're going to break. You put too much energy in, you're going to break some of those bonds, and then all of a sudden your structure starts to unfold. Okay, so changes in these environmental things can denature your protein. It can break your protein. Okay, a denatured protein is considered biologically inactive. If it doesn't have a 3-dimensional shape, it generally doesn't have a function. Okay, so it has to be folded up to do what it's supposed to do. Now, if the primary structure is not lost, if you do not degrade the protein or break the protein so much that you can keep your primary structure, your amino acids attached to one another, you can renature the protein when the environmental conditions go back to normal. Okay? And so that's something that's going to play into some of our labs at some point. I want you to keep this in mind, that we have talked about this, that we have talked about protein structure. okay, and what can cause protein structure to break down. All right, let's briefly, I'm only going to briefly go over these functions of the proteins because you can sit and you can read the purple words. I'm basically just going to read the titles of each one of these. Because there's a lot of functions of proteins. We're going to see a lot of different functions of these things as we continue through the material, and you'll notice them when we get to them. One of the biggest things that we see proteins used for are as catalysts or enzymes that help speed up or facilitate chemical reactions. It's huge. We spend an entire chapter on how enzymes work and how they speed up chemical reactions. We see them in terms of structural materials. A lot of body stuff inside of us is made of protein, right? Your actual muscles are made of protein. The protein that's found in your hair and nails is called keratin. That's a structural protein. Collagen is a protein that's found in your connective tissue. I show this picture, okay, from some before and after, maybe possible collagen injections. That's kind of a thing in Hollywood. Shows what some people will do to kind of change their appearance. People get collagen injections put into their lips. I think both of these young ladies were relatively attractive beforehand, and I don't know, they did something to their face that changed that a little bit at least. But anyway, they did what they thought necessary to make themselves feel comfortable, and you can kind of see that each one of them has had something done to their lips in terms of some type of injection. It might have been Botox, it might have been collagen, it could have been a subtle combination of both. Okay. Proteins also function in specific binding. We see proteins as integral parts of our body's immune system. Antibodies are protein-based. They are proteins that identify foreign substances and basically mark it for destruction. So if you have overcome COVID or know somebody who has done such, their proteins are working overtime. They can also act as specific carriers in many different aspects. We're going to see proteins act as carriers in all sorts of contexts in our material. These pictures illustrate specific binding. This is an antibody. These are the antigens that bind to them, and these proteins are very specifically shaped. The reason that this yellow protein binds to this particular antibody is because this antibody's protein is folded in a very specific way. I talked about that specificity before, and at this point here, it is extremely important, right? It helps you fight off diseases. Down here illustrates membrane transport. We see a lot of these different channels and gateways in our cell membranes that allow certain things into and out of our cells, and they're made up of proteins. So we're going to talk a whole chapter on that as well. Contraction. We see our muscles are made up of proteins, and so they help with our muscle contractions, and also we utilize proteins in signaling. Okay. So there are pictures of some muscles, the fibers interacting with one another. Those muscle fibers are protein-based. They are proteinaceous, if you will, and they work to help us move. And there are certain molecules that act as signals. Insulin, for instance, is one that would be proteinaceous. We see also some hormones might be protein-based, and certain hormones are going to interact in our bodies in certain ways and help us signal certain things in our body. Okay. All right, that'll do it for carbohydrates and proteins. I will be back with another video addressing lipids and nucleic acids. Thanks a bunch, guys.